All right, let's dive into something really cool today. We're going to explore a student project that asks a pretty wild question. Can we give plants a kind of microbial superpower? We're going to break down how some bacteria can act as amazing growth boosters, and it all starts with an idea that's literally right under our feet. I mean, just think about that for a second. What if the next huge leap forward for agriculture wasn't some giant new machine or a super complex chemical, but was actually hiding totally invisible in just one handful of soil? That's the powerful idea we're digging into today. Okay, so to really get why these microbes are such a big deal, we have to start with the main problem. And, like any good detective story, this one starts with a victim, our plant, facing a hidden but very real challenge. And here it is. This is the core problem. Soil is absolutely packed with iron, and plants need it to be healthy and grow strong. But here's the catch. Almost all of that iron is in a chemical form that plants just can't use. It's like being locked out of a pantry that's completely full of food. All that nutrition is right there, but the plant simply doesn't have the key. So if the plant doesn't have the key, maybe something else does. And that's where the investigation really begins. Researchers went looking for microbial helpers right there in the soil. So it's time to bring in the lineup of suspects they found. So this is like the initial rap sheet for our 3 bacterial isolates, MT1, MT2, and MT3. You can see some basic lab tests give us our first clues. MT1 looks like it's from the Bacillus group, while M2 and MT3 are probably in the Pseudomonas family. And that's really exciting because both of those groups are known to have members that are rockstar plant growth promoters. We're definitely on the right trail. That, just being part of the right family isn't enough proof. We need to know if any of our suspects have the specific superpower we're looking for, the one that can solve that iron problem for the plant. So it's time to put them to the test. And that superpower has a name, siderophores. Seriously, the best way to think about a siderophore is like a tiny, custom-made key. The bacteria make these little molecules and send them out. They hunt down that locked-up iron, grab onto it, and then bring it back in a form that the plant can finally use. They're basically the ultimate iron delivery service. So how can you tell which bacteria are the best key makers? Well, you use this really elegant and visual test called the Cas assay. It's awesome. You just put your bacterial suspects on this special blue plate, and if they're making siderophores, they start pulling the iron right out of the blue dye, which creates this amazing yellow halo around them. The bigger and brighter that halo is, the better that bacteria is at unlocking iron. Simple as that. And boom, the results are crystal clear. I mean, look at that. All 3 of our suspects actually tested positive, so they can all make these iron chelates. But while MT1 and MT3 were pretty good, M2 is our undisputed champion. It's not even close. It made the biggest, most vibrant halo, proving it is an absolute siderophore-producing powerhouse. Okay, so we've found our prime suspect. MT2 has the superpower, no doubt about it. But can it actually deliver when it really counts? It's time for the ultimate test, putting this microbe to work with a real plant to see if it can make it grow bigger and stronger. And wow, the results are just stunning. You've got the regular control plant on one side, and on the other, the plant that got treated with our star bacteria MT2. The treated plant grew way more, it developed a much bigger root system, and it had a higher overall weight, or biomass. This is it. This is the smoking gun we were looking for. So let's just boil it down to the most important thing: more biomass. At the end of the day, whether you're talking about farming or just nature, that's what matters. More plant. And our tiny little microscopic helper delivered in a very big way. Case closed, right? We found our hero. Well, in science, it's almost never that simple. Just when you think you've got it all wrapped up, there's a twist in the story. And in our case, it turns out our powerful new friend is still, in some ways, a total stranger. So yeah, the experiment was a huge success. But there's this one critical piece of the puzzle that we still don't have. And it's the one piece that separates a really cool lab experiment from a safe, effective product you can use in the real world. And that missing piece is all about biosafety. See, we know MT2 is probably a Pseudomonas, but we don't know the exact species. And that's so important because while tons of bacteria in that family are helpful, some of them can be opportunistic pathogens. So before we can even think about using this as a widespread biofertilizer, we absolutely have to know exactly what we're dealing with. Responsible science demands it. So, this isn't really the end of the story. It's more like the beginning of the next chapter in the investigation. The work keeps going, and the next steps are actually pretty clear. So, what's next on the docket? First, you've got to get a definitive ID. Scientists will use something called 16S rRNA sequencing. It's basically genetic fingerprinting to get the exact species name. After that, they'll redo the experiments just to make sure the results are solid and repeatable, and they'll run even more tests to build a complete profile. It's all about being thorough, being rigorous, and most importantly, being safe. Which brings us full circle, right back to that big question we started with. This project just brilliantly shows the unbelievable potential that's locked away in the world right under our feet. It's a great reminder that sometimes the most powerful solutions to our biggest problems aren't things we create in a factory, they're things we discover in nature. So it makes you wonder, what other secrets, what other amazing allies are just waiting to be found in the next scoop of soil?